Hello everyone, I am Tareian Cazares, a graduate student in the Immunology PhD program at the University of Cincinnati. My mentor is Dr. Emily Meraldi, a fantastic systems biologist. Today I'm going to be talking to you and introducing you to the Python package our lab has been developing called maxATAC. Transcription factors are DNA binding proteins that directly interpret the genome. There are about 1,600 transcription factors in the humans, and with hundreds being expressed at any given time in any given cell type. There are two main characteristics to transcription factor, the primary one being that TFs have a DNA binding domain. The other characteristic they have is that they regulate transcription through some sort of mechanism. Transcription factor binding is traditionally determined experimentally by ChIP-seq. In ChIP-seq, chromatin bound by TF is fragmented, sequenced, and mapped to the genome to identify transcription factor binding sites. Unfortunately, experimental characterization in all tissues and contexts is currently not feasible. And that's because there's a lack of specific antibodies for transcription factors. There's a large cell number requirement, especially for rare cell types that are often difficult to obtain in humans. And that optimization time and costs are often high and specific for cell type conditions. The largest collection of TF ChIP-seq in a single cell line was published for 208 TFs in HepG2 just 2 years ago. Alternatively, instead of experimental determination, approaches that use chromatin accessibility information can predict many transcription factor binding sites using sequence motif preferences. The current fastest growing data sets are available for ATAC-seq experiments. ATAC-seq is the assay for transposase accessible chromatin, and it measures chromatin accessibility. Compared to other methods of chromatin accessibility measure, it requires much less starting material and it requires much less preparation time, and is also available for single cell. The current standard method for predicting transcription factor binding using ATAC-seq is to first find the regions of accessible chromatin not bound by DNA-bound proteins, such as nucleosomes. Then you scan those sequences that are accessible for known occurrences of the sequences that TFs prefer to bind to, called motifs, in the form of position weight matrices. In this case, the gamma motif is found in these two locations. However, when we compared the standard approach of motif matching for 60 transcription factors and the popular cell line GM12878, we found that actual recovery of experimental transcription factor binding sites was poor. That is a median of AUPR below 0.1. The poor performance of transcription factor motif-based predictions has also been noted by the community at large. In 2017, the ENCODE Gene Challenge pioneered state-of-the-art methods for predicting transcription factor binding sites using additional genomic information. The top performing methods for this challenge increased performance to a median AUPR of 0.4 by using chromatin accessibility information from DNA-seq and mapability tracks. Despite new methods being developed, why aren't state-of-the-art transcription factor binding predictions widely used? Well, it's simply because models don't exist. If you make predictions using transcription factor motifs, there's a catalog of about 1,200 known human motifs available, compared to the 12 transcription factors that can be modeled by state-of-the-art methods. Therefore, our group developed maxATAC. maxATAC is a method that uses DNA sequence and ATAC-seq signal to predict transcription factor binding in a new cell type. maxATAC models are convolutional neural networks that are trained on paired ATAC-seq and ChIP-seq data. Using this approach, we were able to develop models for 127 transcription factors. Our training data consists of 20 cell lines with paired ATAC-seq and ChIP-seq for 127 transcription factors. We started our model development by curating data from ENCODE and GEO databases and additionally generating ATAC-seq replicates for 3 cell lines. In total, we were able to build 127 unique human transcription factor models. For the 74 TFs for which we had at least 2 cell types for training and 1 for independent testing, we compared our performance to the standard approach of motif scanning. For all TFs considered, maxATAC outperformed motif scanning. Additionally, maxATAC made predictions for 5 TFs for which there are no known motifs. With the increasing popularity of single-cell ATAC-seq datasets, we also wanted to test our performance against motif scanning in single-cell ATAC-seq pseudobulk populations. We used an experimental dataset that had 7 cell lines for which we also had bulk ATAC-seq in our dataset. We used models trained on bulk ATAC-seq to make predictions in these 7 cell lines and found that across every comparison except for one, maxATAC outperformed the standard practice of motif scanning on these single-cell pseudobulk populations. Next, we wanted to know whether our models could be used to predict transcription factor binding sites in primary cell lines despite being trained on transformed cell lines. To do this, we used published ATAC-seq and ChIP-seq data in human CD4 T cells that had ChIP-seq available for 3 models in our database. We then compared our maxATAC predictions to published ChIP-seq experiments. We found that for all 3 transcription factors available, maxATAC outperformed motif scanning for genome-wide prediction tasks. The precision of our model at 10% recall was also comparable for most TFs. We next showed that maxATAC can be used to study transcription factor binding in complex disease settings using publicly available data sets for patients with atopic dermatitis and their age-matched controls. CD4 T cells from atopic dermatitis patients or age-matched controls were isolated and activated with anti-CD3 and anti-CD28 for 5 hours. ATAC-seq, whole genome sequencing, and RNA-seq were performed on these samples. For this analysis, we used donor-specific DNA sequence and ATAC-seq to predict transcription factor binding at disease risk loci for atopic dermatitis. With our maxATAC models, we were able to identify 2 transcription factors, MIB and FOXP1, that showed differential binding predictions at 2 haplotype blocks associated with disease risk loci for atopic dermatitis. For more detailed information, please visit poster W053 or our recent bioRxiv update. All of our code and our data is available on GitHub, PyPy, and Docker Hub. All of our data for training our maxATAC models is available on Zenodo.